So yeah, thank you, Garen, for inviting me to give this webinar and for organizing everything. It's actually super nice to see so many people interested in single molecule RNA fish. We have been trying to kind of promote this method among the plant community, but somehow, as far as we know, there were not many people using the method, but maybe I'm wrong, or maybe this is about to change today. But anyway, let's see. Maybe many of you are already using and have many questions for me today. So the method is called Single Molecule RNA FISH, or smFISH. And as the name already suggests, the novelty is that you can really, and I hope you can detect single molecules of RNA. And this is what is already illustrated here in this first image. You can see some small dots that correspond to molecules of RNA here in the cytoplasm. There's also some in the nucleus, and there's some special ones here as well that I will tell you about in a minute. As Garen said, this is kind of a bit of a practical webinar, so I will tell you a little bit about the method. So I'm going to divide my presentation in two parts. In the first part, I will explain you what is smFISH. I'll tell you a little bit about the history and how the method was developed. And then I will tell you some of the important steps within the protocol. So I'll explain you more or less how it is performed, not much into the detail, but just things that I think they are really important. And then I wanted to give you some examples of how or what you can use this method for. And here I decided to actually give some examples from my research, mainly things that I have done working on the gene FLC. So most of it is published work, but I think it's a good collection of experiments that will show you how you can use this method to really ask different types of questions. So I'm talking to you today as an expert in the method, but unfortunately I have not developed it myself. So these two persons here, Rob Singer and Raj Arjun, actually developed the method. The method took really a long time to develop as well. So the first reports where they have shown mRNA detection in single cells were actually done already back in the 80s, in 82 for the first time. And then I have here in 86 where it made the front cover of Cell. The thing with the method then is that it was using very long probes and radioactive probes. And the problem was that in this case, the signal kind of diffuses, and therefore you don't really get, even though you get detection in single cells, you really don't get single molecule detection. You can see in some cases there are all these kind of accumulations in the cytoplasm. You cannot really assign them to single molecules. Another issue with the method as it was then is that with these radioactive probes, it really required super long exposures. In fact, I think some of these images would be exposing for things like a couple of months, 3 months even. So I mean, if I already complain about long exposures, this was a different level of patience. Nevertheless, I think they were amazing, actually, images. You can really see already where the RNA is being accumulated within the cells. So you can see, for example, here, it's not everywhere in the cell, but being accumulated at the growing edges of these cells. So I think it was already pretty amazing that they got that back then. Now, the method actually became like a single molecule detection later in the late 90s, mostly due to advancements in fluorescent microscopy. So this is also work by Rob Singer. And so the difference here was that the probes were basically now labeled with fluorescent dyes. And in this case, they actually managed to have the RNAs having enough accumulation of fluorophores, and in this case, they were forming diffraction limited spots. So it was single molecule already back then. The problem was, however, as you can see, maybe in this image, there was a lot of variability in the signals. And this is mostly what they think it was due to self-quenching of the fluorophores. So it was really difficult in a way to know whether a certain spot really corresponded to the RNA they wanted to detect. And there were issues of specificity as well. And these probes were really hard to synthesize and purify. They were heavily labeled with fluorophores. So then not many people were using this method. So 10 years later, in fact, Raj came in and colleagues and did just a slight tweak on this method that actually worked really well. And that was basically to use now much shorter probes that were, again, directly labeled with the fluorophore. And in this case, just one single fluorophore. And that really improved it in many ways. First, the probes were more easy to synthesize and purify, so they are now commercially available from Stellaris Biosearch Technologies, and are the ones that we've been using. And you had a much more homogeneous signal between the different molecules, so it was a much more specific labeling as well. So this is the method that we have basically so adapted to plants. So what me and Susan did was to basically use it in plants, and so far it's mostly working in roots. I will tell you a little bit about that later. But in fact, even though the method was there, it took us quite a long time to develop it. Plants are often not so simple in terms of microscopy compared to other systems. Mostly due to autofluorescent issues, complicated cell walls, and so on. So it did take us quite some time to get it to work. And I'm very grateful that actually Susan was incredibly positive about it and always believed that we were going to get there at the end. And she was absolutely right. So Susan is also still working a lot on this method. She's been doing some optimizations. So she's also a very good person to contact with questions. I'm sure she's going to be very happy to help anybody interested in this method. So now the question that you may be asking is, how do you know this, or what is special about this method that makes it able to detect single molecules? And this is mainly to do with the way that you probe your RNA. So in the case of the probes that we get from Stellaris, so developed by the group of Raj, they are very short probes, between 18 and 22 nucleotide long. And as I said, they are directly labeled with the fluorophore. Then what you do is that you design them such that they tile along your target, your RNA of interest. So what you're doing then is really concentrating and packing a lot of probes and fluorophores into these single molecules, such that this then generates what is called a diffraction limited dot, okay, these little dots in the cytoplasm. Now, of course, you don't want to go on amplifying the signal too much, which happens in certain methods, because if you amplify it too much, and especially if you have a very, and maybe not even too highly expressed gene, is that you're basically going to stain all the cytoplasm. So all these molecules here that you see nicely individually is just going to come all kind of a blob, and you're going to lose the single molecule ability. So you don't want to amplify too much, but you don't want to also go too little, because of course then you don't see anything. So what Stellaris really recommends is to go up to 48 probes maximum, but they don't really give you a minimum. So I guess it really depends on the system, on the targets as well. In our case, we have managed to detect things with as little as 25 probes. Or even 20, but I mean 20 is really borderline. You already don't have a very good signal, which means that you have to get a very good prep. So there is indeed, as you can see, a limitation of size. So your RNA cannot be too small, because then you're not going to have enough space to fit in all these probes. Now, okay, another thing that you may be asking is, okay, if I go back, is, okay, this still look quite big to me. How do you know this don't really correspond to accumulations of RNA in the cytoplasm, for example? So of course, the people that have developed it thought a lot about these things and did very important controls. And here I would like to mention just some of them. So in this case here from Sanjay's lab, what they did was to really synthesize a bunch of RNAs in vitro, okay, and then label them with single molecule FISH probes in different colors. And then they basically combined them and then injected them in cells. Of course, this cannot be done in plants. So then what they asked was if what you're going to see is yellow dots corresponding to green and red RNA, or if they could see discrete green and red dots, which then would point towards the presence of individual molecules rather than accumulations of RNA, and that's exactly what they saw. Another thing that can be done in order to prove that these are single molecules and not accumulations is to simply look at the size of these dots, which is pretty constant, not only between your samples, but different systems and different organisms as well. And we see similar sizes than determined in other groups. And also to look at the intensity of these dots. So that's another thing that anyone can do, is to basically see what is the intensity and then plot these intensities. In principle, they should give you a unimodal distribution with a very narrow peak, showing that you don't have really different kinds of accumulations of RNA. Now, one place in the cell that you do often actually see accumulations of RNA is in the nucleus. And that's actually quite interesting, because what happens is that these accumulations normally tend to correspond to sites of transcription. During transcription, what you have is that in many situations you have more than one polymerase transcribing the locus. So then you can have several RNAs at your locus, and therefore you get, I mean, in this case, it's not even a big blob. You can have much bigger signals. So you know, for instance, that this cell is actively transcribing at that given moment. If you are really interested in transcription, another thing that you can do is to do something even more specific, and I'm showing in the next slide, which is to label, instead of label or probe your exonic regions, is to label the intronic regions of your RNA, okay? If you have, of course, a big enough intron or enough introns in your gene. So in this case, the gene is PP2A, is just a housekeeping gene. And you can see here, we were able to detect quite nicely just now signals in the nucleus really corresponding to the sites of transcription. Okay, so now here with this probe, we don't detect any signal outside of the nucleus, because, and in fact, they correspond to the locus because splicing really does happen anyway, in most cases, co-transcriptionally. what is really nice about this is that, of course, when you're looking, for example, in this image here, you have all these dots. You may not know which dot correspond to the site of transcription, but because now you can put it together with the intron signal, you know that that's the one here, right? So what you can also do is now compare the intensity of this dot with the other dots, and you can even know how many molecules of RNA you have at your site of transcription. So this method also gives you that kind of information. Okay, this all is looks quite nice, but how difficult is it to get this to work? And here are the good news. I think this is probably one of the easiest methods that you can possibly try. I mean, it's so easy that it's almost embarrassing. I mean, once we got it to work, it really is a very robust method and really not complicated. You can do it in 1 or 1.5 days. And I mean, it's super simple. I really like it, actually. What you have to pay attention to mostly is your probes. You have to get them right, of course, and your material. Really important, but I guess that's kind of general rules for any kind of in situ hybridization that you would do. So your probes, you get them from Stellaris, and they have a software for designing the probes, so it's also quite simple to use. You just paste your sequence on it. It really works more or less like a primer designer, and it will look for characteristics such as GC content, and also try to avoid predicted regions that are highly structured on your RNA. And at the end of that, you will get a list of probes. So at the end, you have to see how many probes you have. Do you have enough probes? If you don't have enough probes, you may go back and just change slightly things in the way the probes are designed. Mostly you can change the size of the probe a little bit and the spacing between the probes as well. And if you do get a good number of probes, then you can select your fluoro4, which actually they give you many options, but be careful there. Don't go with the fluorescent one. We did try that one. It actually wasn't good, and one of the reasons why we were stuck for such a long time. So Quasar 570 and 670 are really the best ones that we recommend for people to use. Now, your tissue, of course, so the issue with the tissue is that even though it looks quite bright in this image, these nice little dots, in fact, they are quite dim. Often, if you just look down the microscope, you don't see them. You do need your relatively good camera to be able to really detect the signals. So if your autofluorescence is high, you're not going to see any signal. So at the moment, the method works really well in roots. In the beginning, we were having an issue with, and still with the whole mounts, and we were doing root squashes, and it's still our favorite method because you get single layers of cells, and that really helps because if you have a very thick tissue, that is of course going to increase autofluorescence. But we have now managed with some clearing methods, not complicated things, to get it to work on the epidermis in whole mounts. And also we have other tissues like embryos. We have managed like early some shoot apical meristems. And I do think if there's more people working on this, eventually we're going to manage to get in many more tissues. I think in leaves is going to still be challenging, but who knows, you know? I think these things, they just need a lot of optimization steps. So hopefully some of you are already trying some of these things. Okay, so of course, very important, especially if you're new to the method, and in general, but I say especially if you're new to the method, is to do your proper controls, because the tissue without any probes is already going to show some sort of autofluorescent speckles that may look like signal. And I have seen this in different, I don't know, people that have shown me examples of that, and they were not looking at the right thing. So really important, do all your right controls, get to know your sample, how it looks like without the probes, and then of course all the other controls. Probe in the absence of your target, so you know you're not targeting something else, ideally a mutant. But if you don't have a mutant of your gene, you can at least also do an RNA treatment. That's not going to tell you about specificity, but at least it's going to tell you about stickiness of the probe to your material, to your tissue. So that's also important. If you are very worried about specificity, one thing that you can do is to use 2 probe sets for the same RNA. So what people normally call the odd and even probes. So you do like green and red, and then you should see pretty much colocalization in all these kind of dots. So that's another control that can be done. Okay, then the protocol itself is actually, as I said, it's really quick and it's very simple. Basically, you fix your sample, you wash. There's one step of ethanol that is actually quite good because you can store your samples at that point, so that's quite convenient. This step is used to permeabilize cells. And then after that, basically, you probe your samples, a couple of washes in the middle, and then the hybridization can be as short as 4 hours. We tend to prefer overnight, but it can be done in a very short time. And because then you don't have to any amplification steps needed, you're basically, again, a couple of washes and you're ready to go. So yeah, really simple. Now the imaging. The imaging, actually, I think is quite important to mention here, because I think people in a way have been put off trying this method because they think, oh, this sounds really complicated, it's single molecule, I may need a super resolution. But in fact, you can go, and it's the best actually, to go with the wide field microscope. You can do it in a confocal, but in fact, it's much harder. So only if you're very experienced with a confocal, then it may work, and it does work, but it's much easier to get good signals and bright signals with a wide field microscope, that then you can use a deconvolution after that. And the only thing that may be important to mention is that you do need long exposure times, okay? So we are using a couple of seconds, depending on the probes and on the targets, 3 seconds, sometimes more, 2 seconds minimum. If you have an EMC CD camera, you can go a bit lower than that. Okay, so now you do your imaging, you get your nice pictures. What do you do with it? So why do you get the single molecules? What is the advantage? So one of the important things is to really quantify. So you want to know how many mRNAs there are in your cells, but you don't want, of course, to count them by hand. So we have been collaborating with computer scientists that actually developed a software for segmentation and automated counting of these mRNA dots. So this is available online, and in fact there will be a new version of this coming soon. Alternatively, if you have MATLAB, you can also use another software that actually has been developed by the group of Raj. It's a lot more friendly, the platform, than the one that we use. But the problem with that one is that you will have to do most of the segmentation manually. So if you have a lot of cells, that's kind of that's going to take you some time. Okay, so then so that was my first introduction to the method. I hope that kind of I covered most of the things. Now I wanted actually to tell you how we have been using this method into research. And as I said, I wanted to tell you a bit about the work that I did while I was a postdoc in the lab of Caroline Dean. So I want to keep it quite simple. As you probably know, important developmental transition in plants is flowering, the transition to flowering. And this gene, flowering locus C, is an important regulator of this step. There are many pathways that control this gene, and vernalization is one of them. So what is vernalization? Vernalization is in fact the perception and the memory of the winter that allows plants to synchronize their flowering with the favorable conditions in spring. For instance, plants would not want to be flowering right now here in Sweden. So what vernalization does, in fact, is it switches FLC off, and this then allows flowering to occur. So it releases the repression, flowering can happen. So many things are known about this gene. It has been widely studied. And I just wanted to have this very simple cartoon here to just say a little bit what I just said, but with a bit more information. So what you have is that the gene is active in the warm, and as I said, in the cold it gets switched off, and it's actually a progressive repression. So it's quantitative and is more and more repressed as with the longer time in the cold. And then in the warm, when plants are back in the spring, the expression does not go back up again. It remains low. This means the gene is epigenetically silenced, and this happens through very complicated pathways, mostly involving polycomb repression. So one of the questions was, why does that take such a long time to switch off a gene? And why is it this quantitative as a function of time? That was a very interesting thing. Why it happens in this way? So a lot of research has been put into this, and actually involving a lot of modeling, and we know now that this is because in fact the gene works as a bistable switch. So it exists either in an on or an off state, and in fact this graded downregulation that we see here, for example in this northern blot, is in fact given by the progressive switch into off state of individual cells. And this is what is shown here. This is FLC tagged with Venus in roots, and with increasing time in the cold, what you see is that there's here some cells being switched off, and there will be more and more cells being switched off with increasing time in the cold. Now, we wanted to understand what is it that switches the gene off in this cell autonomous way. What does switch it off in individual cells? And one of the candidates was this long non-coding RNA. So the FLC can be transcribed in the antisense orientation, okay? And that gives rise to long non-coding RNAs. There's different forms of it, but basically this happens during this cold period. So during the first 2-3 weeks of cold, there is a huge increase in antisense transcription, and we were really interested in understanding whether this increase in transcription could somehow explain or help in this cell autonomous switching mechanism. Now, of course, this is a long non-coding RNA. We cannot really tag with GFP. We want to understand its behavior in single cells. We really needed to have tools for that, and that's how we got interested in single molecule RNA FISH. And of course, we didn't start with this. long non-coding RNA. We, of course, first started with the easier one with this FLC sense, and we developed different types of probes for that one, targeting, as I said before, exonic regions to detect the mRNA, and the intronic regions to detect the nascent RNA. And it looked really nicely. So we were very happy to see that the method was working. Now, of course, we wanted to know, does it behave in a way that we expect as well? So we did, as a proof of concept, just a time course of FLC in the cold using this method. And very nicely, we could see there was less and less RNA in the cells with increasing time in the cold, just as we see by qPCR. And we quantify that. This is just number of mRNAs per cell in these histograms and the frequency of cells. So here, most cells actually already don't have any mRNA. And also quite interesting was to see that the cells where we were targeting the unspliced transcript or the nascent RNA with intronic probes, we see that most cells here at 2-3 weeks were already showing no signal whatsoever. So transcription stops quite early, but you can still detect mRNA after that. So the mRNA has a quite stable half-life in the cold, so it's quite long-lived. But transcription had already stopped much earlier. Okay, now the question was, can we now use this method to detect a long non-coding RNA? So despite its low levels in before cold, we could detect it already then. So this is a nice thing about this method. So because it is a single molecule detection, even if you have a very low expression, if you're looking in the right cells, you're probably going to see it. And this is what we detect, because qPCR almost doesn't detect it before the cold, but here we were looking in the right cells, and I'll come back to that, why I call them the right cells. We could already see some cells with cool air signal, very specifically. Now, what we observed in the cold was an increase in the number of cells that were now transcribing cool air. And this is what I have here quantified. So this pretty much looks with the same kind of behavior that we saw by qPCR, so with a peak around 2-3 weeks. But what was really, well, first, the fact that there were more cells was new, but also what was surprising was to see now that there were bigger signals, and I have here just zoomed in some of these nuclei, bigger signals and also additional signals in the nuclei. So this was totally unexpected. And this is one thing that I really like about this method, and I think about microscopy in general, is that you can, I mean, there are things that you just don't know until you're looking at your cells. So of course we were like, oh, this is really exciting. What does that mean? So the first thing we did for trying to understand the origin of these dots was a sequential RNA-DNA FISH. I have to admit, this is tricky, and you cannot combine these methods. They are very different and have very different hybridization conditions. So we had to do it sequentially, which is difficult, but if you're not looking at hundreds of cells, it's possible. And what we observed was that the big signals were always where the gene was. Okay, so the smaller signals were somewhere else in the nucleoplasm. And we were like, oh, this is maybe very interesting. Maybe cool air is regulating other genes in the genome. So has trans activity rather than, and also a cis potentially. So we looked at other genes. We did double labeling. I put just here one example. And we also looked at potential proteins, because we thought that the pattern in the nucleus reminded us of, in this case, Cajal bodies. So we did immunostaining as well. So you can combine, this is actually something quite nice about this method. You can combine smFISH quite nicely with immunofluorescence. So these protocols are very similar in terms of the conditions that we work. So we did all of this, but in any case, we didn't see any colocalization. So that was a negative result, but that was okay. At least we knew that wasn't the case. So we kept thinking, and we then realized something. And this was that this additional foci could potentially be just the intronic sequence or the intronic lariat, because our probes were in fact targeting the intronic region of cool air. So we wonder if these foci were actually the full-length transcript. So to answer that, we just simply design probes for the exonic regions of cool air, and saw, in fact, that when we do that, we no longer see, this is here in the green, we no longer see the additional foci. So you only see the foci when you're targeting the intronic region, and I have here another example. So what does that mean? So we don't really know what that means, but potentially it could be that the intron is not being degraded, of course, in the locus, maybe because there's so much transcription happening during the cold, during this initial cold period, that there is just too much RNA coming, and then you cannot degrade fast enough, and therefore the intron is just dispersed in the nucleoplasm, awaiting degradation. Or maybe these guys really have an additional function, but we really don't know that. But this is what we figured out so far. Now, these big signals were actually quite interesting as well. I mean, most people have been kind of skeptical about cool air and can we nicely detect it or not. So we were like, okay, it seems to be, when it's being expressed in the right cells, it really goes really full speed, and then you have a lot of signal. What is that actually? We were wondering, are these accumulations of RNA or just bursts of transcription? So we tried to use smFISH to answer that, and here we did this trick. I mean, we were lucky that there's a very long intron, so we did two sets of probes, one in the 5' end and the other one in the 3, so in red and green. Well, in fact, this is far red, but for the purpose of this presentation, I'll call it green. So what we saw when we did this kind of labeling is that you always had, okay, not always, but often had a stronger signal when you were labeling the 5' end of the RNA, and a bit weaker signal when you were labeling the 3' end. And this is even more evident in the second picture here. So what this means, maybe this means this, and I'm explaining here in this cartoon. So what you have is probably several polymerases transcribing the gene at the same time. So here the gray blobs. So you have several RNAs being transcribed at the same time. So you have several binding sites for probes in the 5' region and a bit less in the 3' region. And therefore, why we see often this situation with a bigger signal on the 5'. But every now and then, we saw also cells that showed big signals in both probe sets. And this here in green at the 3' end, it's really a very big signal, so it cannot be a single mRNA molecule. So we think what we have here is both situations, is there's a transcriptional burst, but also the RNA stays at the locus for some time. Maybe there's some stickiness to the locus, but there are definitely some accumulations of RNA at FLC. So this was all kind of new, things that we did not expect to see, that we wouldn't have learned otherwise without this method. But now coming back to our initial question, how is cool air helping in this repression, in this cell autonomous switch? So now we had actually tools to look at transcription with single cell resolution and even single locus resolution. And we tried that, and what we saw was, first, they can be transcribed in the same cells, which was really good news if we think one is influencing the other. But also what we observed was that they were never transcribed from the same copy at the same time. So there was never colocalization. So, and this is what I have here quantified. So we wondered whether this mutual exclusive behavior of cool air transcription in relation to FLC sense could actually explain or help in this process of repression. So we were lucky in a way that we understood that cool air, and this is what I said in the beginning, was not transcribed in all cells. And this is again something we did not know before we tried this method. So somehow cool air is mostly expressed in the stele of the root. We don't really know exactly why that is, and mostly close to the tip of the root. So we could actually ask, what is the repression or the down regulation of FLC in cells that don't transcribe cool air, so like these cells here, versus cells that do. So in cells that don't transcribe cool air, there's like 20% reduction of FLC transcription after 2 weeks in the cold. So there is repression. So there are definitely mechanisms that are independent of cool air that act on this repression. But what we observed was that in cells where cool air is transcribed, this repression is then, of course, I mean, there's a mutual exclusive behavior is much stronger. So this was a really nice result and a kind of an important evidence that the transcription of this long non-coding RNA is relevant for this environmentally triggered epigenetic switch. And another important result that came out of this was, of course, this mutual exclusive behavior. So when we actually represent things like FLC in this way, this is somewhat inaccurate. And what we have is mostly that the gene is being transcribed in one orientation or the other at any given time. Okay. Now, what does that mean? And could this be that there is a mechanism that is trying to really avoid this kind of situation where you're transcribing from both strands at the same time? Or is it we see this behavior just because of a probability effect? Let's say the probabilities of initiation are so low that they never transcribe at the same time because the probability is just low. Now, we could in fact use this method to determine this kind of probabilities to understand what are the initiations, the rates of initiation, and many other rates, in fact, from FLC transcription. And we did that again together with computer scientists and mathematical modelers. And we extracted a whole number of transcription parameters from FLC, from Pol2 elongation, transcription initiation, intern processing, and so on. And I'm just going to briefly give you some examples here. So one thing that we noticed when we wanted to do this was that we thought we had a problem because there's a lot of variability in the number of mRNAs per cell. And this is what I'm showing here. So there are cells with as many as almost 200 RNAs, okay, and cells with 10. Okay, so if you're trying to determine rates with such a variability, it makes no sense. So you were like, okay, actually there has been many reports of genes showing this kind of very variable and stochastic transcription. So in a way, it was maybe not so surprising, but it could have been a problem for us in order to determine those parameters. We wanted to understand where was the variability coming from, where was the noise coming from. Was the noise coming from the level of production of the RNA or at the degradation of the RNA? And to do that, we started with the easiest option here, the degradation, because to study degradation, we could just simply stop transcription using drug treatment, in this case, actinomycin D, and then actually look to what happens to the half-life of the RNA, how quickly is degraded, is this variable, is it not? And also a good thing is that you could use smFISH to really accurately know whether the cells had indeed stopped transcribing. So you can see here, so this is before the treatment, you see transcription sites, and after that 4 hours, we were sure that there was no more transcription happening. So there was only decay of RNA, and this is what we looked at here. And what we observed, and I'm not going to go into the detail, was that the degradation was pretty constant, and the half-life of the mRNA could be set to 6 hours. So that means that the noise was then coming at the level of the production, and that could have been a problem. But we also noticed something else. So we noticed that bigger cells always had more mRNA. So we thought, okay, let's see what is the relation between the number of mRNAs to the size of the cell. So we went back on our data and determined the cell volumes for the cells where we had already determined the number of RNAs, and plot them against each other, and saw this really nice correlation between cell volume and number of mRNAs per cell. So what this means really is really super interesting. It means that the cells, they know how big they are somehow, and they adjust transcription to maintain a constant concentration of RNAs in the cell. So now it does make sense to determine initiation rates, because we can just set our analysis to a certain size of cell. So there isn't all the variability that we thought there was in the system. So we determined all these kind of rates, and we could finally go on and ask what was indeed the probability of initiation from both promoters. Or in other words, what was the probability of having 2 polymerases transcribing the gene at the same time? And in theory, of course, we realized that would be around 20%. So that means that in our smFISH experiments, we should be seeing 20 out of 100 cells showing some sort of colocalization signals, which we totally don't observe. So this isn't happening somehow. Why this is it, and how the cells are controlling this, this is something that I'm now really interested in understanding and verifying as well for other genes. And with that, I actually just wanted to finish by summing up what you can actually learn with this method that you wouldn't learn from a bulk analysis such as by qPCR. So first thing that this method is super useful for, and as I showed you, is to really quantify the number of mRNAs per cell. So you can really count the number of dots, and you can know in absolute terms how many RNAs are in your cell. You can study noise in transcription, variability in gene expression, and so on. Another thing is really important, of course, you have a single cell detection ability. So you can really accurately know what is, in which cells your transcript is present. So here, no GFP, so you know the endogenous RNA where it is, and maybe you will be surprised, like we were, to know in which cells it appears. You can then, of course, look at the subcellular localization of your mRNAs. So here you have a very uniform distribution of mRNAs in the cell, but in many cases isn't like that. So we have seen cases where the RNA tends to be, in fact, accumulated in the nucleus. It can go into some kind of compartments inside the cytoplasm or bodies. So this is something that other methods will not tell you. Then if you're really interested in transcription, you can, as I mentioned, use the intronic probes to really study active transcription. You can know how many copies are transcribing at any given time. You can, in fact, even know how many RNAs you have at your transcription sites. And you can study different types of transcriptional states, such as bursty transcription between other modes. You can determine all kinds of transcriptional rates, elongation, splicing, initiation, and you can really accurately study mRNA stability. And I guess with that, I come to my final slide already. So of course, I need to acknowledge a lot of people. I have not done this on my own. I have to thank a lot Caroline Deans and Martin Howard groups that really allowed us to kind of adventure ourselves into this method, and that actually have been really supportive in our work. And still today, I still benefit a lot from collaborating with them. Then, of course, Susan, that I already mentioned, for really being my partner in crime in establishing this method for plants. Matthew and Jelvar, that did all the automated detection and image analysis. And then Uja and Robert for all the work that we did on the transcription parameters. And finally, Angelo Valeriani from the MPI in Germany, from whom I did the latest simulations for the double pole to occupancy. And finally, also, my still very new and small group, Anis and Shue, but very enthusiastic group, so that I have also embraced this method and that are already doing it even better than me. So, and of course, funding for allowing us to do this. INDEPTH, that has also been really great and a good platform to really share this method, and from which I have benefited a lot from interactions with many groups. And everybody for being present here today, and I will be happy to take questions.
Thanks very much, Stefanie. So thanks very much for this really detailed in exploration of smFISH, and I especially like you obviously you went back over your published data, but you went to it in real detail that maybe you wouldn't get unless you really delved into the paper. So that was really interesting to see. So we have a few questions come through. So first of all, we had one from Francesca Lopez. She sent it in previously, and she's asking, so the example you use is just in root tissue. Have you tried to use it, and why can't you use it? And if not, why can't you use it in other tissues?
Yeah, so as I mentioned, I mean, the main problem is autofluorescence. So I've been working up till now mostly with roots. I do a lot of microscopy, and roots are always my favorite because mainly of they are just much easier for microscopy. So this is the main reason. But if you work with any other tissue that is not highly autofluorescent, as most plant tissues are, then it may well work. So, or if you have a good protocol for clearing. But it's not all about chlorophyll. There are other things in plant tissues that just glow and that makes things more complicated. Therefore, that's why we started with the roots, but even the roots were hard. So that gives you an impression of how hard it is.
Fair enough. With everything, you have to give it a go, and if it works, it works. So we have a question from Anna Puna Alu, and they ask, what's the typical length of a probe that you use?
The typical length, 20 nucleotides. So they give you not much margin for trying different things. So between 18 to 22 nucleotide, but yeah, so very short probes. But generally, you would use 20 nucleotide.
So I guess a follow-on question from that. So you're obviously using these probe sets from the company. Have you got how much do they cost? That could be an issue for some people, I guess. So.
Yeah, they are expensive. One thing that I can already advise people, if you are new and going to try this for the first time, I would recommend, because of course, if they are expensive, they are like 500, 700 euros a set. But, and then you're like, okay, I don't want to buy it before I know it's working. I know that it is possible to contact them and ask for a couple of sample to try, and we've been in touch with them a lot, so I think they would probably give you a couple of microliters from our control probe, so you could just try in your lab and make sure it's working for you before you invest some money on it.
There we go. So you would, if you hadn't come to this webinar, you wouldn't know that little tip there. That's
right.
So a question from, we got quite a few questions actually, so it's really great. Thanks everyone. I'll try and get to all of these. So from Risco Hadi, and they ask, how did you go from the smFISH images to quantifying and abstracting those? So from the individual images to get out the data.
Yeah, so we collaborated with computer scientists. I wouldn't be able to do that myself, but they have actually been developing the algorithms for automated segmentation and automated detection of dots. So they are free, so you can just follow on the link that we have in the paper. I also put here on the presentation. I can put them, I don't know.
I put that link in the chat for anyone. So the GitHub link is in the chat. Yeah, so ultimately they're image analysis people, and they segment things and they look for dots. Doesn't matter what dot it is, it's dots, right? So dots and dots.
Yeah, exactly. Yeah.
Okay, so a question from Daniele Cordiero, and they ask, do you think the method could be used for microRNA detection?
Actually, as it is, no. I have seen people use, well, there was one paper that they use variation of this method for microRNA, but not this one, because the problem is, of course, the size. So you need, as I mentioned, at least, I would say 500 base pairs minimum. But people have now been developing ways to amplify the signals. We have not tried a lot of it, but we've been trying some of these new methods, and we have not yet succeeded, but I'm sure we will eventually get there. So if you're really interested, I would look into it. Yeah.
Okay, cool. Okay, so questions from Clara Richard Borbus, and so she has a few questions actually about FLC, but one which is quite technical asks, does ethanol perturb the nuclear organization? She says she doesn't see any chromocenters in any of your pictures. So have you got any thoughts on that?
No, I don't think the ethanol is perturbing the chromocenters. I think probably what you have is, most of my images are from the meristem. And in the meristem, what you have is like a massive nucleolus. So most of the nucleus is the nucleolus, and the chromocenters are not so visible in these cells, I would say. But we have seen them in the more differentiated cells without any issues. So I think it's just a cell type situation.
I think that's fair enough. And kind of a follow-up from that, or a related question from Clara about ploidy levels. So can you see any correlation between cell size, number of molecules, and the ploidy levels? I'm guessing in those meristematic tissues, they're all like 2C cells anyway, so maybe
yeah,
that's
they
not
were mostly 2C. I mean, we cannot for sure guarantee there's not some endoreduplicated cells in the cells that we have analyzed, because yeah, at some point within the meristems, there's already certain degree of endoreduplication already starting at more kind of late meristem, but most of the cells would be diploid, the ones that we analyze. But I'm sure that that would be very interesting to look, because of course endoreduplicated cells will be bigger cells as well, and see how that would then scale up. Transcription would scale up in the cells.
Mm-hmm. So a question from Ronell Roth, and I don't know about this, so hopefully this makes sense to you. So how does your method compare with ACD's RNA scope? Do you know what that is?
Oh, RNA scope, yes. It's a very similar method. It's very simple. There are many similarities, but RNA scope does have an amplification step. So it uses some kind of smFISH probes, but it does, so what it does, instead of using, so these probes are 20 nucleotide and they are specific to your RNA. What RNA scope does is it makes, I don't remember exactly the size, a couple of nucleotides specific to your RNA, and then it has a tail that is a common tail between all the probes they use. Now they use this tail to then add some smFISH probes into their tails and amplify the signal. So this in principle indeed could help for transcripts that are much shorter, but has a specificity problem. I think Susan has tried, if it was not RNA scope, was from a similar kind of method, and I think she struggled to see that her probes were showing the same RNA, so there were some non-specific dots there. Mm-hmm. So yeah, that's all I know about it. I have not really tried it myself. I guess you can try to optimize these things in order to kind of remove all these non-specific signals. I think it could be interesting, this method as well, the RNA scope for genes that where you cannot use smFISH.
And I put Susan's contact information in the chat as well. So if she gets bombarded, we should write to her and say, if you get bombarded with a bunch of questions. Yeah, that's true. But so question from Ortran. She asks, do you ever see an effect on secondary structure of the target RNAs? Because treatment at 37 degrees might not resolve all this structure during hybridization.
Yeah, I mean, we really thought that we would have a problem with that, but it doesn't seem to be the case. I mean, cool air is highly structured itself. Mm-hmm. And people have looked from other systems to hairpins, like in the MS2 system, and they still managed to get the signal very nicely. So somehow the structure, either the structure is not a very stable structure, so there's some kind of changing from conformations, and this is enough for RNA, the probes, to come and bind to some molecules. Maybe that could be one thing, that these things are not like super stable. But the fact is that we are detecting signals on very highly structured RNAs without doing any stringent denaturations or anything.
Mm-hmm. So kind of related to that is a question from Christophe Tatou, who you may be familiar with. So he asks, how many genes have you tested so far? So obviously the published stuff is the PP2A, the FLC, the cool air. And have you done any other stuff in the lab since then?
Yeah,
Okay. So
we have done many things, and I have to say that I almost struggle to think about things that didn't work.
Okay.
So we have used cell cycle markers, for example. We have used other genes, many things, in fact, that have worked. So.
And all within the kind of root meristem tissue, is that way?
We have been, yeah, of course, we've been looking at things that we know that should be expressed there. But yes, so far we've been using focusing on the root, because that's where it's working best. Mm-hmm. But we have already tried other tissues, as I said.
Yeah, some hints towards publications coming there, hopefully. So a question from Kalyana Krishna. She asks, so how do you select the fluorophores? Are they pre-designed with the probes? And do you see any limitations in this? So you mentioned about the fluorescein in your talk. Does she says, are there any limitations about like bleed through or different probes and things like that? So
yeah, that's why I suggested those two, because they are the furthest ones you can have. The other ones will give you some sort of overlap. So that's why we would go with the red and far red, the most far red they have. And they have other ones, of course, you can use. We haven't really tried them, but I would not recommend the green one. And yes, when you order, basically you select with fluorophore and they come coupled with a probe. So I don't know if that was, did I answer?
No, I think that's it. Yeah, absolutely. So you can select there. That's good. So question from Ulkar Ahamdi, and they ask whether you think they can use the method to detect the level of transcription. They work with a highly transcribed gene. So do you think you could use this method for that?
For detecting levels of transcription?
Yeah, level of transcription.
Yeah, exactly. This method is really beautiful to study gene expression in single cells. So not only gives you the level, but it gives much more than the levels. Gives you the level per cell, and gives you the single cell resolution and subcellular localization. So, but you can really count number of mRNAs per cell. So it gives you, I mean, you don't even really, like in qPCR, you don't need just to normalize your data, because pretty much what you get is what you have. So you get the number of molecules. And then you can learn about all these variabilities between cells and so on. So.
Yeah, that's true. So a question from Isabel Coles, and Isabel asks whether you can use it on isolated cells, so kind of, I guess, protoplasts maybe. And she also asks if it's compatible with immunolocalization. So you mentioned it is in your talk. So in that case, though, do you just put the antibodies in at the same time as you put the probes in? How does that work? So
yeah. So to answer the first question, yes, I mean, this is why we've been squashing the roots to really kind of get isolated cells. So if you're working on isolated cells, it's really good already. It's an advantage. You don't have to worry about like a thick tissue and the autofluorescence that comes with it. It could be that your cell is very autofluorescent and it's maybe a problem. But yes, it should work in single cells. And yes, people have combined the immunofluorescence protocol in different ways. They have done first the immuno and then the smFISH, the other way around, and combining, and it seems to be working in all possibilities. And also you can do a sequential as well. So I don't think there is a big restriction on how you do it. I would recommend myself doing that. This is the way we did first, the RNA first, because we thought that would be I don't know, more likely to go wrong somehow, and we wanted that one to be right. We were also working with immunofluorescence with a very good antibody. So that's the way we tried, and I have not tried in other ways, but I don't have a special reason to think that the opposite direction wouldn't work.
Yeah, okay. So yeah, this kind of follows on from a question about the microRNA. So from Asmar Sultana asks whether the probes are kind of similar length of small RNAs. So do you worry that there could be the problem with cross reaction with those on the basis of complementarity? If a gene produces those small RNAs, could you be just picking up that, do you think?
Yeah, this is the thing, is like one molecule doesn't give you enough signal.
Of course, yeah.
Yes, really, I mean, this is a very good thing about, it can be also considered a bad thing if you're trying to detect. But no, it's in general a good thing, because what you have is you need a lot of probes in the same place to visualize. So even if your probe is binding to something else, which most likely it will do, it will not give you enough signal. So you will have to have many of them together to be able to detect the signal.
Yeah, of course, the devil's in the detail that you look into it, you think, oh, there's just one or two probes, but there's 48 probes, so you get a huge amplification with that, obviously. So that's important. Yeah. that's a good point. So a question from Ahmet Tech. How do you differentiate between RNA, RNA, and RNA DNA hybrid formation? Have you tested that, and do you need to test them to ensure that it's an authentic?
That we are not detecting DNA.
Mm-hmm,
yeah. We actually been trying to use these probes to detect DNA, and it actually is extremely difficult.
Okay.
So we don't think it is a problem. In fact, I don't think we are detecting the DNA at all.
Okay.
So yeah.
Okay.
Try to think about which controls. I mean, of course, when we do the RNase treatment, there's no RNA in your sample, and then you have only the DNA. You're not detecting the DNA. And then we have been trying to. I've been trying with Sophie when she was here for STSM to really do this denaturing to get the probes in. But somehow it seems to be extremely difficult to either, I don't know what it is, to kind of denature your DNA in this specific sequence, because you need that specific sequence to be all denatured and get your probes there. But it really, it seems really complicated to get this to work. I don't think no one ever, actually, I haven't seen any reports of that.
Okay, so maybe just a couple more questions. So question from Ronelle again. So she asked, to quantify transcript levels, do you need to normalize your messenger RNA against an internal control as with qPCR?
No, as I said, so basically you just get the number of molecules, and yeah, so you don't really need any normalization. Of course, if you're worried to see, okay, is this behaving in a way that you expect, you can. We have compared the dynamics of the gene with qPCR data, but you don't need any internal normalization. That makes sense.
Yeah, okay. Yeah, I mean, yeah, you use control anyway. So so this is a question more about FLC biology. So I know you moved a little bit away from this, so you might not be so familiar. So this is from Alvaro, and he asks, how does the FLC protein levels behave on vernalization? Do you have the same level of resolution as you do for the RNA?
If we get single molecule protein, I didn't get it. So how the protein?
So just in general, yeah, what happens to FLC protein after vernalization?
So of course, I mean, if you stop transcribing, as we also show, I mean, by smFISH, but also by qPCR and other methods, eventually you will run out of RNA, you will run out of protein. So there is a decay of the protein. There's, of course, the levels are going down. We do see this bistable switch in a way that if you wait long enough, that cell, there's going to be a decay of the protein. So it's just going to, you will have no protein on the cell anymore, which will take a couple of hours depending to the half-life, which I don't know what is the half-life of the protein, but.
Yeah, I mean, that's the majority of the work in Caroline's lab is all about the incredible complex regulation of the FLC mRNA, and I think so, and the protein levels kind of follow on from that, I guess. Yeah. Okay, so I'm going to final question then. So this is from Clara again, and she asks whether you know if translation might interfere with probe hybridization, and highly translated molecules might be harder to detect in that way.
Yeah, people have looked into so if ribosomes are interfering.
Yeah, okay.
Yeah, so people have looked at this by labeling the untranslated regions of RNAs and compared that with the coding regions, and they don't seem to have to see a difference. So they did again different, as far as I remember, they did different colors, and they see colocalization. So if the binding of the ribosome would be an issue, that you would see many cases where you wouldn't see a colocalization, but that's not what they saw. So we don't think that's a problem. And
the fact you have, again, 48 probes along the entire transcript, you know, you would think if it's busy at one point, then another bit would give a probe, maybe a signal.
Could be, yeah. But we don't have to do any protein proteinase treatment or anything, and we just detect a lot of it. So for many transcripts, so
okay, great.
of course they are there to be translated, and if we're still detecting them, I don't think it's a problem.
Okay, I think we'll leave it there. Well, that's, I mean, thank you very much, Stefanie. So I will say to everyone still on the line, so we still have a good number of people here. I mean, some of you will have come to my the webinar series, the UK Plant Science Presents webinar series, and we struggled to get questions on that one. So I think this is really shown that this sort of technical thing has worked out so well in this first instance, that people have really come out and asked these, you know, there's some detailed questions there, and Stefanie has been able to answer them all pretty well, I think, Stefanie. Yeah, so I think it's been really successful. So as I said, you know, huge thanks to Stefanie for this, and I'll do some editing with this, and I will put this. This will be online on the INDEPTH YouTube channel. Discuss with Stefanie exactly what is needed there. So you know, you can use it as a resource and send it to your colleagues as well in the future if they have any questions about that. I think we've had a really great introduction to both the technique and then kind of some of the difficulties of analysis of the sense and the anti-sense, different transcripts and things. So it's been a fantastic talk. So thanks everyone for attending, and thank you to Stefanie very much again for giving us the information.
Thank you, Garen.